Right, this lesson is about genes and it's topic 3.1. Here are our syllabus points. There are quite a lot of them, so make sure you have a read. All right, genes are a heritable factor. They are inherited from parent organisms. They are a strand of DNA, don't forget, so our nucleotides made up of nitrogenous bases with a sugar phosphate backbone. Now, they are influential to specific characteristics of an organism and they stem from that protein, which is coded for by the DNA strand. It's a small part of the DNA strand, a gene, okay? Genetics is the field interested in how that information is stored, expressed, and passed on in family lines. All right, genes in our species, even though we are very, very complex creatures, we are not the organism with the most genes. We code for we have about 20,000 protein coding genes in our genome. Here's our classic BioNinja image, and we can see that we are just less complex there than the water flea and rice. Okay, this is just talking about the number of genes that we have. It doesn't necessarily refer to complexity of so many other elements in our body. All right, genes occupy a specific position on a chromosome, remembering that chromosome are long strands of DNA and they are tightly wound together and around proteins. So one specific chromosome has that gene, okay? It is not spread across, it is just on one spot, and that's usually if there's no abnormalities, okay? It can shift around if we start to see abnormalities. They are labelled according to their position on the chromosome, and you can see here we have a centromere that sits generally in the middle where that pinching point is. And our numbers, if this is the X chromosome, our numbers start to get bigger the further away from that centromere we get. So the top arm is the P arm, the bottom arm is the Q arm. So linked genes can actually be multiple genes that work together, and they might be across different chromosomes, but remember, one gene sits in one specific spot on one chromosome. Alleles are specific forms of genes. They are different versions of the same gene. So that's like saying, okay, I've got an entire recipe book full of recipes for soup. One might be tomato, one might be pumpkin, one might be minestrone, but they are literally all for soup. Okay, that's what alleles are. They are different forms of the same thing. So they are found in the same place on the same paired chromosome. So it'll be on if you remember, you've got pairs. So if you're talking chromosome 7, you've got 2 of them. You've got 2 copies, one from mum, one from dad. So one paternal, one maternal, but they code for a slight variation on a protein. So in this particular example, you're talking about allele for white flowers and an allele for purple flowers. So that organism carries both, and which one it expresses, we'll talk about down the track. So you might carry a gene for brown hair and a gene for blonde hair, and depending on which one is going to be expressed is depending on what your characteristic looks like, but they still code for hair color. Okay, so alleles can differ only by maybe 1 or 2 nucleotides, not huge at all. In this particular case, if you look at the DNA, GGC and its complementary CCG, it's got a single nucleotide polymorphism. Poly meaning many, morphism meaning shape. All right, so it's got just a little slight variation on what it's presenting there, and it's changed to GAC. And that might actually just be one tiny different, the brown hair to blonde hair situation. All right, bioinformatics analyzes and interprets all of this biological data we're talking about and all the genes we've spoken about. So online databases allow us to compare variants in genes so we can see what's a normal variant, what might actually be a negative mutation. Now, we often associate the word mutation with something that is negative, but that's not always the case. They can be actual beneficial, they can be neutral, and they can be a negative thing, as we've said. So the alleles, right, those differences in genes and the single nucleotide polymorphisms, they're versions of mutations, okay? They're just sort of normal mutations. Not all of them are bad. Some can be invisible. So for example, if you have a look at this, we've changed from TAT to TAC, but what that's actually done is literally nothing to the amino acid. If we think back to our mRNA and amino acid table, you know that multiple codons coded for the same amino acid, and in this case, that's what's happening. So there's not much of a change there going on at all. If, however, we can compare this here, and this is the string of amino acids, we've changed from a blue one to a red one, you might actually see a change in that protein. It's still going to be the I pigment, but it's just a different version of it. So some mutations can be about the change in nucleotide, that single nucleotide change, and some can be the change in the whole codon, so the 3 nucleotides there. Some of them can happen de novo, which means spontaneously, and some can be induced by other things that are mutagens, like the sun and UV damage and things like that. So if we're talking single nucleotide changes, we can have a substitution where we've changed this T straight to a C. You can have an addition where something is actually slotted in, and what that's going to do is change everything downstream from it, or something, a nucleotide could be deleted, and once again that's going to change everything downstream from it again. These mutations cause variation, which is fantastic for a species and allows it to evolve. So a really good example of that are the peppered moths. They are a European species, and as the Industrial Revolution began, more of the machinery and the pollution started to hit the atmosphere, and what happened was the lichen on the tree, which is this light stuff here, it starts to get killed off. And previously, the lighter versions of the moth, the lighter variants, it was good because they could camouflage into that light lichen on the tree. But once that lichen started to die off, it's not really beneficial to be a light moth in a dark background. So natural selection starts to favor the moths that can camouflage. So previously it would have been the light ones. Now moving on, it would be the dark ones. So the light ones getting picked off as prey and eaten, and the genes are being passed down by the dark ones that could camouflage. All right, lethal mutations can occur if an organism's gametes, so the sperm and the egg in our case, they inherit something that means that that gene is not letting that organism survive. And it's a way to weed out DNA errors and potential problems. Often these lethal mutations are found in genes for growth and development. So it's kind of nature's way of saying, well, we know this isn't going to grow, so let's not continue trying to grow it at all. And this is a Wikipedia image just showing that, say, this gene is inherited from both parents. That organism is not going to survive. All right, some point mutations, so those one base changes. Some point mutation disorders include sickle cell anemia, cystic fibrosis, and color blindness. So one change in one tiny nucleotide and we get massive issues. Sickle cell is the example we're going to talk specifically about, and it's a point mutation that changes an A to a T, okay, an adenine to a thymine on the DNA strand. And what happens is the transcription and translation occur, and it changes the hemoglobin protein that is made. Hemoglobin, remember, carries oxygen around in the red blood cells. So if the amino acid has changed, then the protein changes, and the change is actually in the shape of the hemoglobin molecule, and that also then changes the shape of the red blood cell to make it look like a sickle. This is a sickle, it's an old gardening tool. So once the shape changes, it makes it difficult to push that cell through the capillaries, the really tiny blood vessels, and patients have red blood cells that are susceptible to breaking. Now, the shape is being pushed around as it gets pushed through the blood vessels, so they also have a shortened lifespan, the red blood cells. So this is HL content, but I still think it's really interesting. The rates of sickle cell anemia are higher in people of African, Middle Eastern, and Indian and Mediterranean descent. And these are all areas where malaria can also be endemic. So interestingly, people with sickle cell anemia can be immune to certain types of malaria. The malarial parasite has part of its lifestyle inside the red blood cells. So if the shape is changed of that sickle cell, it means that the parasite can't grow in there, and they burst and too early and the parasite can't grow. So in some ways, sickle cell anemia can be considered a beneficial mutation in reference to malaria. All right, our genome is the whole of our genetic information in the organism. So every single one of those nucleotides, and we can compare that to proteome, remembering all the types of protein that can be created by the body. In eukaryotes, it's all the DNA in the nucleus, plus there's mitochondrial DNA. And in prokaryotes, it's the DNA floating around inside that single cell organism, as well as some plasmids, which you'll learn a little bit more about later on. Now, the Human Genome Project sequenced about 3 billion nucleotides from 23 chromosomes. And if you want to talk about them in pairs, you can double that up. So it began in the 90s and was a genuinely international effort from scientists, which is pretty rare. Usually they're quite competitive. So the first draft came out in 2001, so more than 10 years later, and the final came out in 2004. That's 3 years of drafting. As technology improved, then the project could as well. So it was a genome. It wasn't the genome, it was a genome, remembering that there are heaps of variations between me and the next person and the next person. So don't forget, just because we know the general sequence of what the Human Genome Project has actually found, that doesn't mean we understand what it all means yet. Okay, and it's really important when it comes to ethical issues of biotechnology. We don't know what it means. Should we declare it We don't know yet. Okay, so and also one scientist junk, so we don't need this bit, we don't care about that, it's not really a coding gene, might actually be another scientist's treasure. Okay, so until we know what it all means, we can't be sure that, you know, what we're doing is the best technology or anything like that just yet. All right, quite an important unit there, lots to look at there in the syllabus points.